Hello and welcome to this special program on Doordarshan. I am Taposh Bhattacharya, and in this special program we take you on a ride of the Indian Ocean with this team from CSIR's National Institute of Oceanography.
23 members of CSIR's National Institute of Oceanography are on a mission. The 90-day mission that has the potential to change the way the Indian Ocean is studied and viewed. This is their story of grit and determination in adverse conditions to unravel the mysteries of the Indian Ocean.
The 90-day scientific cruise mission is expected to bolster India's research in the field of commercial biotechnology application, along with an understanding of the climate change and its impact on the Indian Ocean. These scientists will be working day and night to unravel the mysteries of Indian Ocean, and we get you the details in this report.
One of the key objectives of the mission is to identify and characterize the genes and proteins in the ocean to understand the cellular level processes occurring in the microorganisms' response. Proteins act as markers and catalysts for the biochemical reactions occurring in the organisms existing in various conditions in the ocean. By studying proteomics, one can understand the various cellular biochemical changes and their response to climate change, nutrient stress, and increasing pollution. By studying proteomics, the researchers will be able to identify the physiological responses of the organisms under varying ocean conditions. This will allow the scientists to understand cellular biochemistry and the response of the ocean to climate change, nutrient stress, and pollution. Trace metals affect marine life, the driving force in ocean waters and sediments. Their importance in marine ecology is undeniable. Hence, a thorough understanding of the factors affecting marine ecosystems is essential. Conceptualized over the last 2 years, the project will take 3 years to complete. The group will be cruising through the Indian Ocean for nearly 3 months, collecting a huge amount of samples to unravel the mysteries in the Indian Ocean. The sampling will be carried out over a period of 3 years under the CSIR-funded project TRACEBIOME.
Ocean occupies 70% of the global area and controls global climate. It is very much important for us to understand the processes which are acting in the ocean, to understand the global climate, how it is changing, what are the stress factors, how the pollution is controlling, how the temperature rise is controlling, how the pH change is controlling, how global climate change and ocean warming is changing the biology of the ocean. To understand this at cellular level, National Institute of Oceanography, Goa has started a big program to understand the biogeochemical study or process at cellular level in the ocean. What we plan to do is to measure genomics, proteomics and trace metal like micronutrients and all in the Indian Ocean, in the different parts of the ocean like seawater, sediments, biology, forums, diatoms, and even microbes, bacteria. Every places we want to study this, and we want to go to the cellular level. So what we are planning to do is to collect the samples in entire Indian Ocean and to analyze them for RNA, DNA, trace metal study and various proteins.
We have been working on this formulation of this project for several years. Although NIO is working in this field of Indian Ocean, looking at it in a different perspective with all the modern technologies available was a must, because Western countries have already done many of this kind of work in Pacific and Atlantic, and Indian Ocean, in spite of being the third largest ocean in the world and the closest to India, has really not worked upon. And that is where NIO and Government of India with the different institutes should step in. And we've looked into formulation of this project where we wanted to explore the vast openness of the Indian Ocean and what is lying in the water column and the sediment.
From ancient time, Indian Ocean has been used as a channel for transportation, but much of it is still unexplored. Now 23 scientists and researchers on board RV Sindhu Sadhana are trying to unravel the mysteries of Indian Ocean. These scientists and researchers are working to understand the impact of climate change, at the same time how trace metals plays an important role in marine diversity.
What is trace metal? These metals are supplied to oceans via continental runoffs, atmospheric deposition, hydrothermal activities and continental shelf interaction, and they are essential for ocean productivity. Trace metals are considered as non-degradable pollutants that will accumulate in coastal environments, mainly in sediments, to levels that might be toxic to the biota. Their toxic properties result from complex interactions between numerous biotic and abiotic factors. The understanding of this complex mechanistic phenomenon therefore requires both laboratory and field-based toxicological studies with adequate model organisms. The adverse effects of trace metals pollution in contaminated environments can be substantial and long-lasting despite years of restoration efforts. For these reasons, the monitoring of the contamination by trace metals remains an environmental issue of great concern, not only for the biota, but also human beings. In the changing scenario of climate, it will be interesting to look into the naturally occurring trace metals in the Indian Ocean and trace their source. With the advent of modern sequencing and analytical tools in molecular biology, it is important to study the effects of trace metals on marine biota and their metabolic responses to existing levels, along with possible future elevated levels.
What we call as TRACEBIOME, and which is being funded by CSIR for the next 3 years. So this is a 3-year program. It's not only 3 years, we would say this is initiation of that program what we'll be looking into, and we will try to link the biology with the trace metals. Now coming to this expedition, which we have started and embarked upon, say it's one of the longest expeditions without touching any port in between, and all the participants staying on board continuously looking at different aspects. The main objectives of this expeditions are to look into again the trace metals and the different kinds of nutrients which are available in seawater, which are going to affect, but all these days are speculative, like okay, this might happen, that might happen, which we will be doing a ground truthing in this voyage, wherein we will look upon different kinds of microorganisms which are present, different kinds of planktonic life forms which are existing therein, and how they are getting affected by these metals or the nutrients, and how the climate change might be. This will be basically draw a baseline of the data which we are looking into establishing in this project. The different aspects which we are going to cover in this, as I said, would be the trace metals and different kinds of isotopes, which will be looking into the seawater column throughout from the surface up to the bottom of the water, and also into the sediments, the top at least 1 to 6 meters of sediment which we will be targeting to collect in this cruise. Under the biology parameters, we will be looking into the enumeration of the bacteria or the microorganisms and the plankton. All these years we have been working in, it's not that nothing new for NIO, we were looking into it, but the aspect through which we were looking were quite normal what we are doing. But here we are trying to get into some of the modern omic technologies which people have been using basically for the clinical diagnosis or the other different life sciences field. But unfortunately in Indonesia we have really not made use of these omic technologies. Last 5 years there are few papers and studies which have been talking about that, but it is not a concerted effort where it is one of experiments or studies where person has done basically for one project. But here we are trying to combine and pull everything together, which so the different omic technologies which we are using would be basically genomics, then we will be using transcriptomics and proteomics. When you talk about genomics, genomics will give you who is there. Transcriptome will give you what this who are present are doing, and proteomics will give you the information and how it is being done. So what would happen if suddenly this change or this reduce? So that will be having some simulated experiments on board directly, which will give us a real-time effect of what would happen in future if these changes occur. Now coming to the application aspect of this, it's not that we are going to look into only the basic parameters or the basic sciences to just understand what is happening. Ultimately it has linked to the applications. That is where we will pull in the biotechnologists who are there in our group. We will be doing some preliminary work with respect to that, like for example isolation of bacteria, isolation of fungi, isolation of different plankton, which might give us different molecules which could go into the drug research, which will go into the food research. With the microcosm experiments what you are planning on board, it will give us a fair idea about the climate changes which people are talking about might happen in future, wherein we are talking about the change in the pH or the rising of sea temperature, sea water temperatures, and how it is going to affect the organisms present there. Like although we as human beings are used to fluctuating temperatures of say 10 to 15 degrees during summer and winter, that doesn't happen with the organisms present in the seawater. Even a temperature change of 1 to 2 degrees is huge for them, wherein their physiology and the metabolism, what is present, how they are behaving or how they are behaving under stress changes, and this is linked ultimately to climate change and the climate change modelers, which they will use this information what we are collecting on those crews, and they would be able to predict the models better. In this 90-day expedition, which we started from East Coast at Visakhapatnam port, we plan to cover Bay of Bengal, Indian Ocean along the 30 degrees south transect, and Arabian Sea. And this is the first time where Indian Ocean is going to be covered this way, where we are going to sample Bay of Bengal, Indian Ocean and Arabian Sea in one stretch and one cruise, which has never been happened before.
The mission was expected to begin from the 11th of March from Visakhapatnam port, but the team faced their first real test when two cargo containers still couldn't make it to the research vessel Sindhu Sadhana. The tension was palpable on the face of the crew, especially Dr. Samir Damare, who is heading the expedition. 13th February 2021. After a delay of 2 days, the cargo finally arrived, containing the important equipment including the conductivity temperature depth instrument, but again the team faced a problem as the CTD instrument was found to be not working properly. 14th February 2021. Another day of delay and the faces were dropping, but Dr. Samir kept his composure. Engineers working on board Sindhu Sadhana then came up with an ingenious idea, replacing a part of the instrument with an old one. And the smile was back on their faces. But they still faced the problem of fuel shortage, as they were almost 4 days behind schedule. They worked out a formula to refuel in Port Blair, and thus the journey started for the team to conduct their cutting-edge research in genome and proteome mapping of microorganisms present in the seawater and sediments. The expedition started from the Visakhapatnam port, going to the mouth of Hooghly, Brahmaputra, and going southwards right up to 30 degrees south latitude along 90 degrees east. With a fuel refuel at Mauritius, the team will continue the return journey northwards, coming up to the exclusive economic zone of Pakistan and back to Goa. The expedition will cover approximately 10,000 nautical miles. The team will be cruising through CSIR-NIO's Sindhu Sadhana cruise ship, an 80-meter long and 56-meter wide research vessel. The vessel has a dynamic positioning system to allow holding the ship at a point of various sampling.
This is an electrical propulsion ship. We have 4 generators and 2 thrusters in the forward and aft also. It is called azimuth thruster. The propeller can rotate in 360 degrees at any point of time. So in full, it will give around 13 knots speed. Design speed, presently we will be getting around 9 knots, 8.5 knots. It depends on the weather it changes.
We have 4 generators of 1200 kilowatt, and it is electrical propulsion with 2 azimuth propellers and 2 bow thrusters. So that the maneuverability, it has got a self-maneuverability capacity to go anywhere. And it has got a full power we can give while speed 2 generators, and 2 generators can be standby. And regarding the long voyage like this, NIO plan to have a freshwater generator also, and we have one more supporting to that RO plant also, where we can generate sufficient freshwater required.
15th March 2021. Happiness replaced the gloom that engulfed the crew, who are now sailing into the vastness of the sea. As the vessel picked up speed, so did the NIO crew, who got busy unpacking their equipment and getting ready for their testing before their first stop. After around 3:30 p.m., IST Dr. Samir Damre called for a meeting of all the team members to brief them about the mission. After losing 4 days, their main objective was to get back the lost time, and thus they plotted a new route which would give them the precious fuel and water supply. At around 5 p.m. came the emergency drill undertaken by the Chief Officer Sidney Brito regarding the emergency sirens and the best way to put the life jackets along with other emergency steps. A rapt crew listened to the diktats.
See, basically, the moment we sail out from the vessel, 24 hours prior departure, when the crew change is done, when the scientists are staying more than 24 hours, we have to do a drill, which has been just carried out. The purpose of the drill is we have to be ready in any type of emergency. It can be any type of emergency. We have to be prepared. That's why we have kept all your PPE all in order, your life jackets, your helmets, so that the people are aware of the duties, what is to be done in an emergency. There should not be any panic. So that's why we have explained them the launching procedure of the life rafts, then the launching procedure of the rescue boat and other life-saving appliances which is present on the ship.
16th March 2021. Somewhere near the coast of Paradeep and Haldia port, the vessel stopped. It's time for the first stop and collection of sampling. Everyone was working at breakneck speed, trying to get things working. Technical officers Anthony D'Souza and Kuldeep Kumar, along with a host of researchers, were giving final touches to the equipment which would be used to collect samples at different depths.
Okay, we are in the final preparation of operating for the first time clean CTD in the Indian Ocean. Basically, we have done a dead weight test for the winch, and now the cleaning of the CTD winch is being in progress. So once this is done, then we'll be lowering the full CTD into the water, and that's for the Indian Ocean.
One of the key objectives of the mission is to identify and characterize the genes and proteins in the ocean to understand the cellular level operations of organisms in the ocean. Thus, it will help to strengthen the country's research into commercial biotechnology applications, as well as help understand new climate change patterns. The group, which woke up at around 7 a.m., was working till late night. In fact, some of them had to work the whole night for their experiments. Such was the dedication of the team. The collection of sampling finally concluded on the next morning. By now, everyone was looking tired and jaded, but at the same time thrilled with the way things progressed.
In this cruise, we are concentrating on different aspects of this group of organisms. Okay, because they are primarily important because they form a basic link in aquatic food web, including fisheries. That is the first thing. And second thing is they play a major role in biogeochemistry. The reason is because they are photosynthetic, and they contribute globally almost 50% of the global primary production.
Global?
Primary production.
What does it mean?
Primary production is the amount of carbon dioxide fixed through photosynthetic process. Okay. The third important point is some of the members of these phytoplankton, they are responsible for causing blooms, including harmful algal blooms. Okay, and also some of them also known to contaminate seafood through toxin production. And above all, some of them are also has some biotechnological application because they have different kinds of marker pigments which can be of use in some industries like nutraceutical companies or other, because they produce like beta carotene and lutein and other. But need not say, the thing is the phytoplankton is represented by diverse group of organisms representing different groups, and they are also represented by thousands of specimens under this particular group. Indian Ocean is important, mainly the northern Indian Ocean is most important because it's On the Arabian Sea, consider of the oxygen minimum zone, and Bay of Bengal is differently in most contrast to the Arabian Sea, and much of the studies were focused on these two systems so far, and it was not much attempted to go the southern side of the Indian Ocean. So here for the first time we are taking a cruise which will cover all these systems, the both Arabian Sea and Bay of Bengal, and also the Indian Ocean, southern Indian Ocean system. Then hopefully the first thing is we will be able to have good information on the diversity of this plankton organism because we are going to have live imaging organisms. But traditionally during the voyage the analysis of these microorganisms generally done through microscopy. But on the board it is not possible to carry out the microscopy analysis. That's the first thing. And by the time we complete, it is almost the 90 days. So till then it is not correct to keep the sample preserved then to analyze, because when you add preservative, it is a chemical. So will destruct the organism, some of the organism, not all the organism. So therefore the live imaging analysis will give the actual what kind of organisms are there.
What was quite an eye-opener was to see the way women researchers were putting in hard work and getting ready for the first sample testing. Their grit, determination and steely resolve is what makes them stand apart from the crowd.
So in this cruise, will be collecting water samples for chemical analysis as well as microbiological analysis. Now there are some basic chemical parameters that we sample. For example, first is dissolved oxygen concentration. Now dissolved oxygen, the concentration of dissolved oxygen, it varies depending on the depth, and at each depth the dissolved oxygen concentration will be different, and it might affect the type of microorganisms that are present in that at that particular depth. So that is why we need to know what is the concentration at particular depth. Whatever bacteria or fungi cultures we will get, we'll be doing the DNA extraction here, and then later we'll be doing the sequencing and identifying what organisms are present, and after that we'll be doing enzyme studies.
Basically on this cruise, the work that I'll be dealing with is RNA extractions, which is metatranscriptomic. So water column, I will be concentrating on the chlorophyll maxima, that is around 60 to 100 meters depending upon the kind of waters. If the waters are clear, the chlorophyll maxima shifts lower. So I will be working with the chlorophyll maxima to see how trace metals will affect the microbial community there.
This is my first cruise. I am very excited to learn new things here. I am doing something new here about diatoms. As diatom is the eukaryotic unicellular algae, I am collecting here sample for the diatom diversity and the culturing of the diatoms. When I will culture the diatoms, I choose dominance on this culture and trying to give the different environmental condition to the diatoms, and we'll see what response diatoms gives to that environmental.
Right now I am in the Indian Ocean on board research vessel Sindhu Sadhana. Here I am going to do the concentration and the isotopic studies of the micronutrients like iron, cadmium and zinc in the Indian Ocean. We are away from our home for 90 days. We are not in connection with our parents. There is no internet, there is no phone calls, only satellite phone calls are there. So it's like yes, we are missing our home, we are missing our parents, but still we are very interested to our work. We are very enthusiastic towards whatever we are doing, and yes, there are so many physical challenges are there to be on board. Like every time there is no time for doing work. You can get a station even at daytime, even at night time. There is no certain sleeping time, eating time, but still we are doing it as a team. We are taking it as a challenge for 90 days.
My work includes studying the microorganisms that are there in the Indian Ocean. Microorganisms are the organisms that you cannot see with the naked eye and are microscopic. And of the total microorganisms, there is only 1% of microorganisms that are culturable, especially in terms of bacteria. So the work that I do focuses on the taxonomic profiles, that is we take the water sample or the sediment sample from different depths, and I just go for sequencing the DNA or RNA that I would get from it. Of the 3 biomolecules that are important, that is DNA, RNA and protein, my work focuses majorly on DNA and protein. So in DNA, I would just see what type of organisms are present and how they are different from location to location. While the protein profiles at the same locations, at the same depths that I would be doing the DNA work, would give me what functions they are doing and how they are important in the different oceanic processes like biochemical cycles or nutrient cycling, etc.
Rapid advances in sequencing technologies and bioinformatics have enabled exploration of the ocean genome. Exploring the ocean genome will increase the number of commercial biotechnology applications extending from multiple anti-cancer treatments to cosmetics and industrial enzymes to antiviral molecules. Exploration of the ocean at a genetic level will result in new insights into taxonomy and adaptive capacity that can optimize the conservation efforts. It was exhaustive for the crew as they had not slept for 2 straight days and were doing research and collecting samples to be taken to the National Institute of Oceanography lab in Goa for further research.
Indian Ocean is the 3rd largest ocean in the world, and it helps in regulating atmospheric oxygen and at the same time plays a key role in climate change. An exploration of Indian Ocean at the genetic level will go a long way in understanding the different genetic model in this particular ocean, and also same time in safeguarding and preserving this particular ocean.
In the next episode, we will be talking about the impact of research on climate change and how some of the experiments done on board RV Sindhu Sadhana are not just cutting edge, but also have a far-reaching impact on the study of the Indian Ocean.